will describe the potential benefits of cloning using examples, examine the role of chromosomes in genetic inheritance, determine the structure of DNA, and examine the application of genetic engineering, analyze the pros and cons of genetically engineered crop plants, and analyze how gene therapy benefits humans. So for this week, we are actually talking about biotechnology and benefits and risks of using GMOs. So as far as you are to uh, to go on with our discussion, we will be examining first what are genes. So genes, which are found on chromosomes, control phenotypic traits that are passed on from parents to offspring. So when you say phenotypic traits, these are actually your physical appearance traits, so or characteristics. Example is your eyes, your skin color, and any other physical traits that your parents can pass from generation to generation. So meaning, it's like, for example, your mother have inherited to you your skin color or even the blackness of your hair. So that's it for phenotypic traits. So when these are passed from generation to generation or from parents to offspring, these are actually what you call as the genes. So thus, genes must contain information, a genetic material which influences the biological makeup of an organism. So early on, it was not clear to scientists that deoxyribonucleic acid, or the what you call as the DNA, is the genetic material found in genes. So because long time ago, hindi pa naman nila alam na meron tayong na merong DNA, as to these traits can pass the phenotypic traits. So nung una wala pa yan. Okay, along with that time, so scientists are able to discover DNA, and that is now we are able to believe that ay meron pala tayong genetic material. So it was only after years of more studies that they were able to elucidate or conclude that okay, what are the functions and structure of genes? So because of technology also upgrading, so they're able now, or we are able now to know that there is what you call as DNA. So early studies on the genetic material was conducted by Frederick Griffith. Okay, so his first major experiment that led to the discovery of DNA as a genetic material was performed in 1928. So he was trying to search for better ways to fight pneumonia. So with the search of that particular disease, so he was able to also discover DNA. So in his experiment, he discovered that the heat killed or disease-causing bacteria contained a factor that can transform harmless bacteria into infectious bacteria. So that's why he was able to perform his experiment, and then he found out that there was this factor that can transform harmless bacteria into an infectious one. So siguro when he studied that one, so nakita niyo yung factor wherein that harmless bacteria was able to get infections, and then kaya the later part katong bacteria was able to be infected instead of kanang living na wala pa lang unta tong iyang disease. And then that factor could be what you call as the DNA. Okay, so early studies on the genetic material is also conducted by Oswald Avery. So he was a Canadian-American physician and medical researcher. So you can say that they are also working in a medical research or even mga doctors. And then they made an extract of the heat-killed bacteria. So their initial treated the extract with enzymes that digest proteins, lipids, carbohydrates, and other molecules and ribonucleic acid. So right now, nanapod tayo gitawag na RNA. And observe that transformation occurred, and then tried treating the extracts with enzyme that destroyed DNA, and transformation did not occur. From these experiments, Avery and his colleagues concluded that DNA was a transforming factor and genes are made up of DNA. So with those things that they are able to for the treatment and then wala yung nahitabo, so they conclude that the DNA was the transforming factor and that the genes are made up of DNA. So, okay, another early studies on the genetic material is accounted for Alfred Hershey and Martha Chase. So, before performed another experiment that further strengthened the conclusion that genes are made up of DNA. So, ano man yun ng mga scientists, they're not, if they are not sure with a thing, then they uh, go with further study. So, they can, they can be able to um kanang they will be able to answer those questions kato ilang mga hypothesis and they can support it with their further experiments. So mo na daghan na pud nag study regarding the genetic material as this fascinates their attention. So the experiments involved a bacteriophage, a type of virus that attacks bacteria. So Hershey and Chase were aware that a bacteriophage contains both protein and DNA. So kato na time, syempre, they're able, 1928 patong kay Griffith. So later in time, kanang nana jud to ang existence sa study with the DNA. So kato gigamit ang pud nilang knowledge so they can conclude for another further research of about the genetic material. So they know that that if they could determine which of these molecules enter a bacterium during an infection, they could conclude which molecule makes up the genes of the phage. So results of the experiment confirmed that the genetic material of a bacteriophage is DNA and not protein. So even if katong bacteriophage can able to attack the bacteria, they're able to conclude that in their with their experiment, syempre, that bacteriophage also contains DNA. So to be able to attack with the bacteria. Okay, so right now, okay, meron tayong illustration of this one. Okay, bacteriophage structure with this structure, and then they're able to study the deeper portions of the phage, and then they concluded na meron pa itong DNA. Okay, so we have DNA structure. So for the DNA, so what makes the DNA to function? So mo na another scientist to experiment for that one. So it was P.A. Levine, a biochemist, determined the basic structure of nucleotides that make up a DNA molecule. So he was able to identify this. Okay, we have the nucleotide is composed of a 5-carbon sugar, a phosphate group, and nitrogenous base. So, because he's a biochemist, then okay, biologically in life, what makes it function? So, iya po nag-studyhan. Okay, the chemistry of life. So, one ng biochemistry, they work for what makes up our the living or organisms, systems, kana mga parts, unsay mga chemistry of it. So he was able to find the nucleotide structure or shall we say kanang DNA mo na siyang nucleotide. So mo na yung structure. So nucleic acids, there are two types of nucleic acids. We have deoxyribonucleic acids and ribonucleic acids. So for DNA, mo na ato ang genetic material, RNA, still can also be. Okay, but some of it kaning RNA moreover kanin siya sa mga single-celled organism or mga smaller organisms. Okay, mo na ilang genetic material. Okay, so in 1950, American biochemist Erwin Chargaff discovered that the amount of guanine is equal to the amount of the cytosine, and the amount of adenine is almost equal to thymine. So if you still remember your biology subjects from your lower years, katong mga, when you're still in the, I think, 7th grade, so you're able to know that in DNA, meron tayong adenine, guanine, thymine, cytosine. So for that, it was Erwin Chargaff who concluded for the Chargaff's rule, which is merong for an adenine, there should be an equal or balanced number to thymine and guanine to cytosine, and so on and so forth. Okay, after Chargaff, the scientists who is independently investigating the DNA structure were Rosalind Franklin, an English scientist, Maurice Wilkins, a British physicist, and Francis Crick, a British physicist, and James Watson, an American biologist. So, their findings provided information as to the structure of DNA. So, this time para tayong nagpunta naman ng history kasi we were able to know those scientists who worked or who contributed to the makeup of those DNA, or to the knowledge of the DNA, mga structures or our genetic material, which are very important for also for us, because kung wala sila, okay, we're not able to know, or even dili na to the sign or the present generation right now will not be able to know mga possible treatments for diseases, okay, kung wala ning mga knowledge of the DNA. So we also have biotechnology. So when you say biotechnology, these are the use of living organisms or their products to modify human health and human environment. So we have selective breeding or genetic engineering. So if you are able to see mga news that they get DNA from specific species, for example, mga lahi na animals, and then they breed it for the other animals, then they're not using biotechnology. Okay, tawag na biotechnology, meaning they're going to modify human health for, syempre, studies lang na siya, studies or experimentation, if allowed, or kung pwede ba i-modify ang isa ka particular na species or organism. Okay, but somehow dili man pud tanan pwede. So na may mga laws that they are going to follow, especially the scientists, because they are the ones to do the experiments. So dili lang ta pwede pud maghanap ng mga animals to patyon or even human beings. So kailangan na they can follow rules or laws for that. So, mo na siya, example lang ni is from this one. Okay, we have before and after here. So, if you can see from the picture, this is a dog, but the dog is meron siyang gimodify. So, you can no longer see, okay, or you can see here na merong na change with before and after. So, this is because of a selective breeding modification, and we also have genetic engineering. So we are going to use the DNA structure and then replacement, okay, and then syempre they're going to use technology for that, higher forms of technology. So dili lang tanggalon din diretso sa atong na body, but it's like smaller structures are being replaced, okay, using technology. Okay, so I still think that you're still following with this video discussion. If you have still questions later on, you can actually comment or comment your questions, okay, right after I posted this one. Okay, so we have selective breeding. Okay, unnatural selection is the process of breeding plants and animals for particular genetic traits. So certain traits are enhanced by selectively killing the organisms that doesn't have the desired traits. So examples are the domesticated animals. So for this selective breeding, it's like how will you modify a particular organism, okay, by selectively killing the organism and getting, okay, something, particular genetic traits ang imong gusto for that particular animal, then or organism, then you are going to modify it. So mo na siya ang selective breeding. So if you need, or if the scientist knows that this particular trait is not needed anymore, then they're going to uh, selectively kill the organism. So, mao na siya ang for selective breeding. So, two common methods are hybridization. So, crossing dissimilar individuals to bring the best of both organisms resulting to hybrids. So, when you say hybridization, is like dissimilar individuals, so meaning not the same organisms are being uh, across with each other, so they can give the best organism for both of the species. So, we have example here for the tigon. So a tigon is a male tiger, okay, plus a female lion. So it enables to inherit the traits from a tiger and a trait from a lion. So this is now a breed from a lion and tiger. So we have named deer tigon. Okay, we also have zonkey. It's a breed from, okay, two different species organism. Okay, we have the zebra and a donkey. And hini, so female donkey and male horse. So hybridization with a male jaguar plus a female lion, then it inherit the traits from those two, then jag lion. Okay, we have also roller bear, I think, rather bear. Okay, polar bear and a brown bear. That's a beard, not beer, but a bear. Okay, hybridization with a coy wolf, okay, a coyote, and a wolf. Actually, it's a coyote ata. And, okay, wolfine. Savannah cat, o di makita. Okay, so savannah, no. Wolfine, male false killer whale, and female bottlenose dolphin. Okay, we also have buffalo, plus cow is a beefalo. Oh, meron pang ganito. Okay, hybridization with a narwhal plus beluga. That is narluga. Okay, a camel and a llama, which is a kama. A cow plus wild yak, zoo. And then male leopard plus female lion, it's leopon. So, you can see here that two organisms were being combined with a combined trait. So, we have producing a new type of species. So, they also have inbreeding. So, it continued breeding of individuals with similar characteristics, closely related, use it to maintain desirable characteristics. So, for this one, it's a breed of individuals with same characters or physical appearance and not physical traits. So, that they should be closely related kay para dili pod lain ang iyahang result. So, for that, because with that desirable characteristics, so mo nang nahunahunaan siya og breed. So what are these in breeding? So let's move forward. So in breeding includes sample like this. Here we have same lang na species, which is a dog. Okay, but the family of dogs, if you're able to know, dagan may klase kasi na dogs, no, but still it's a closely related na mga animals, or I mean, the different breeds of dog is closely related, and kana ginabreed siya with the same desirable characteristics, then they can form still a combination of both the parents of the dog. So, and then, nga na ang ma-form. So, if you can see, ayos po ang iyang result. Okay, so, okay, genetic engineering. So, another term that we have for biotechnology is genetic engineering. So genetic modification, okay, direct human manipulation of an organism's genetic material in a way that does not occur under natural conditions. So this one is a manipulation of an organism's genetic material. So meaning we're now dealing with katulad yung mga higher forms of uh, technology para makamanipulate ta og uh, genetic material. So, gina-change na nato ang DNA structure. Okay? So, dili ako. But, the ones who are doing this one. So, meaning, dili lang po, bisan kinsa lang. So, they're studying this one. So, you cannot just manipulate on your own, but through studies and your knowledge of anything, then, pwede. Okay, so under natural conditions, so which we say, meron pa ring natural conditions, so they follow a specific set of rules. Okay, so they cannot also dili nila makaligtaan because that is meron tayong given na laws, so dili lang ka pwede mag-manipulate og isa ka organism without further reading mga international laws kay kabalo na mo with the ignorance of the law excuses no one. So nga na man na siya. So nowadays dili gani ka pwede mag-study or dili ka pwede kanang mga cadaver, dili na nimo siya pwede studyhan lang na di ka mananghid or even dili ka mag-sign of mga papers kay bawal na siya. So mapriso ka ana. So mo na kailangan you still have to study for more and for or not to conduct something na dili pwede. So, for genetic modification, so just right now, even karon para we are able to get kana sa virus na to na problem kana they are going to use mga human beings, 'di ba, as for how to make the vaccine. So kato bisan kato gani kay kailangan pa nila og mga consents ana. So they can modify on a drug or vaccine para sa people or magamit sa tao. So we also have gen splicing, the process in which fragments of DNA from one or more different organisms are cut and combined to form recombinant DNA. So this is now recombination of the different DNAs from one organism to another. So to be able to syempre form another recombinant DNA. Okay, so we have also recombinant DNA is combining two or more sequences that do not normally occur together through the process of gene splicing. So recombinant DNA is the combining of two or more sequences. So in a DNA na siya, DNA structure. So also recombinant DNA is combining two or more sequences that do not normally occur together through the process of gene splicing. So recombinant DNA is the combining of two or more sequences. So in a DNA na siya, DNA structure. So in a process called the gene splicing. So genetic modification with the DNA, so money. So we are now dealing with the DNA mismo. So kung ganina kay katong hybridization or kanang hybridization or inbreeding is like naturally occurring imo na siya gidiretso breed, but this one okay, we're on kanang changing the DNA sequences. Okay, so right now, so dili na ko siya ma-view. Okay, so we have here, okay, a variant, example lang ni siya. So variant of maize that is genetically altered to express the bacterial Bacillus thuringiensis toxin in which is poisonous to corn borer. So for this, kay gikuan ang DNA structure of maize or corn para dili siya they altered the DNA, meaning gi-modify ang DNA sa maize so that later on dili siya maapektuhan sa bacterial growth of poisonous bacteria. So kanimo na ning sample so para maka-grow og corn that is without disease or without a bacteria. Okay, we have also gene therapy. So it is used to correct genetic disorders. So for that, DNA or the gene is being healed. Okay, mo naging na therapy. So if you can see, karon kay ayos naman ang mga technology, forms of technology, unlike before. So kani, so we can actually correct the genetic disorder through kani, this is or the process, harvesting the cells from the patient. So example na siguro ani kay mga cancer cells. So kadto gina-alter ang or gina-change para ano siya, i-heal ang katong particular na katong disorder. So, in lab, virus altered so cannot reproduce. So, kuhaon tong katong cell na or sample cell tapos ginahil nila or therapy, and then altered so cannot reproduce the virus. So, a gene is inserted into the virus, and then altered virus mixed with patient's cell. And then, if you know genetic division or gene division of cell, so mo to para makareplicate siya og another katong walay kuan na gene kanang na change na ang iyang good na iyang gene. So katong nawala na ang virus. So mo na cells become genetically altered, and then altered cells injected into body's patients, and then other cells produce desired protein. So, for this one, for your knowledge of this one, it's like getting the cells from the patient, same cell from the patient, tapos butangan na po siya, change ang katong gene that allows for the growth of the virus. So, para kuhaon to siya, and then ibalik na po tong cell na gikuha sa patient, since katuman na cell kay it can be readable by the body of that particular patient. So, pwede ra siya ingon ana. Okay, sige, but this one kanang true laboratories niha dili lang pud ta pwede kita ang magbuhatan ni. Okay, so cloning. So, you still remember the Dolly sheep that is cloned by Keith Campbell. So we have a clone is a member of a population of genetically identical cells produced from a single cell. So through cloning, skin cells from adult were gikuha, and then parang donor cells nucleus from mammary glands or other. So it is katong giba so fertilize niya ang fertilized egg from female. Okay, true. The DNA nagikuha from the adult clone. And then, so there is the growing of an embryo to the surrogate mother. And then, meron na tayong later on na cloned na animal. So, moto, cell fusion. So, even if wala na fertilized ang katong egg from female, but with growing the embryo to the surrogate mother, then maka-form siya og baby, or shall we say the cloned animal. That is for the process of cloning. So the first animal cloned was sea urchin. So more on unahon jud pag-test sa mga these processes ang katong mga available lang na animal. So if maka-remember mo ang pinakauna nila jud studyhan always katong mga small animals such as rats, nakay, it can be seen kanang available lang siya. So the first animal close was a sea urchin. So getting an embryo getting an egg, and then mag-fuse siya with an unfertilized egg. So maka-form siya og new cell. And then or even embryo. Okay, so what are the risks and benefits of using genetically modified organisms? So possibility of causing allergic reaction. So through this, pwede po siya na risk. So kining GMOs, genetically modified organism, monit ni siya mga gi-applyan og mga technology to modify or manipulate its genes. And then, so syempre na yung mga possible risks. So if it happened that that GMO or that organism is being tested, so possible pwede mag-cause of allergic reaction. And it can also disrupt the natural ecosystem. So natural, meaning na change naman siya, it's now going artificial. So nanay ma-change sa ecosystem. A genetically modified food could cause abnormal conditions or problems for those suffering from chronic illness. So, meaning abnormal condition naman. So, because it's not a, wala naman siya follow natural things. So, possibility of new diseases being introduced into the community. So, yes, it is possible for new diseases. And ethical concerns man playing God by manipulation. So, mo na siyang pinaka-heavy. It's like, kung unsay naana sa ato and then we manipulated. Even if creation is, nga na ang, iba na lang may order tanan for creation. So, if we're going to use technology to manipulate things or modify the organisms, plants, and animals, it's like, dili na siya para siyang may immoral good na we're changing what is given by us by God. So benefits of GMOs. So if it has a lot of risk, it can also have benefits. So higher level of tolerance to various environmental issues. So with this, syempre ang mga scientists, they're going to modify organisms so for the benefit man jud. So that is why they're going to do such things. So, could make certain foods more nutritious with the addition of minerals and vitamins. Yes. And ecologically, possibility of better waste management, resistance to certain diseases and insects, and more crops are produced. So, moreover, these are what the scientists think to for the organism to benefit and even the community to benefit. So that ends my discussion for the perpetuation of life with biotechnology and the GMOs and its risks and benefits. So if you have questions, you may comment down your questions and I'll try to answer that one. So this ends my discussion for this day. Goodbye and thank you for listening.